Welcome to another IBM webinar. I'm Morgan from IBM Technology, and I'm glad you could join us today. It's only been about 1 month since I joined the company, so this is my very first webinar hosting. I'm excited to be here with you. In today's session, we'll be discussing from cells to cancer, intravital imaging of immune responses. We will explore how intravital microscopy captures real-time immune cell dynamics in living organisms, highlights research cases on tumor infiltrating lymphocytes, and show how this technology adds values to the in vivo evaluation of immunotherapies. As you listen to Sowan's presentation, feel free to visit our website, ibmtech.com, and our shop page, ibmtech.shop, to learn more about the imaging tools we'll be discussing. Now, I'd like to introduce today's presenter. Sowan from our technical application team will be leading the presentation. She combines her academic expertise with hands-on experience to support researchers in advancing in vivo imaging. Please feel free to submit your questions in the Q&A box below at any time during the presentation. We will address as many as we can at the end of the session. Sowan, if you're ready, please go ahead with your presentation.
Thank you for your kind introduction, Morgan. Hello, everyone. My name is Suwon Lee, and I'm a technical application specialist at IBM Technology. Thank you very much for joining us today. In this webinar, I will present data obtained using our intravital microscopy system to visualize immune cell dynamics, and then move into a key application area where this technology is used extensively, such as studying anti-tumor immune activation. I will share both representative research cases and the types of data we can generate with this approach. So here is the flow for today's session. First, I'll provide a brief introduction to intravital microscopy. So what it is and what makes it a unique and powerful tool. Next, I'll showcase examples of immune cell dynamics in the ear, skin, and lymph nodes of mice, captured using our IBM system. Finally, we will look at real-time research cases where we are monitored immune cell movement and tumor-infiltrating lymphocytes, as known as TILs, within cancer models. First, I'll briefly introduce what the intravital microscopy is. So intravital microscopy is a high-resolution imaging technique that allows for the real-time observation of cellular dynamics within a living organism. It helps researchers overcome the long-standing limitations of traditional imaging methods. Traditional in vitro or ex vivo approaches, which rely on fixed tissue slides or culture cells, cannot capture the complexity and continuous changes of a living system. While modalities like PET or MRI provide valuable in vivo information, they operate at the organ or tissue level and lack the spatial resolution needed to follow individual cells in action. In contrast, intravital microscopy enables real-time tracking of drug behavior, biological responses, and disease progression at the single-cell level within living animals. It is a powerful tool for visualizing complex biological dynamics with both high spatial and temporal resolution. Then, why is the intravital becoming essential to researchers? In conventional animal studies, researchers often have to sacrifice different animals at each time point to obtain tissue samples. This approach makes it impossible to follow changes in the same subject over time. and it can reduce statistical reliability. However, with IVM, repeated imaging can be performed on the same animal, providing more consistent, reproducible data while also reducing the number of animals required, which is an advantage not only in terms of scientific accuracy, but also from an ethical standpoint. Conventional in vivo imaging devices such as X-ray, PET, MRI, or IVIS provide variable whole body or organ level information, but they are limited to static snapshots or relatively coarse temporal resolution. They can show overall cell or drug distribution, but not the continuous, dynamic process taking place in real time. Intravital microscopy goes beyond these limitations. It offers live, single-cell level visualization within intact tissue, allowing researchers to capture biological phenomena that were previously inaccessible. This includes not only dynamic cellular behaviors in their native microenvironment, but also real-time visualization of how novel biotherapeutics, such as antibody-based drugs, cell therapies, and gene therapies, are delivered to their targets and act within the body. This capability enables true in vivo drug testing directly at the site of action. However, despite its great potential to advance research, intravital microscopy is still not widely adopted. One of the main reasons is the technical challenge of capturing cellular movements in real time. Inside the body, red blood cells and immune cells move extremely quickly, making it difficult for conventional microscopes to track these rapid dynamics without motion blur or loss of detail. At IBM Technology, we have addressed this limitation by implementing ultra-fast scanning technology. In particular, we use both polygon meter and galvanometer meter simultaneously, enabling precise real-time imaging of fast-moving cells. So the video you see here is an example of red blood cells flowing rapidly through the heart, captured in real time. Using a resolution of 512 by 512 pixels at 50 frames per second, we can accurately record even these high-speed movements. In addition to the ultra-fast scanning technology I mentioned earlier, we provide a fully integrated, standardized system for in vivo imaging in live mice. First, to enable longitudinal imaging of internal organs, we have been continuously developing and optimizing a variety of organ-specific imaging devices tailored for different mouse tissues or organs. Because we image organs in a living state, natural movements caused by breathing or heartbeat can make it difficult to obtain clean, high-quality images within conventional microscopies. To solve this, IBM Technology has developed its own motion compensation algorithm, which corrects for these movements and processes crisp, stable images. We also provide systems to maintain the animal's body temperature during anesthesia, ensuring stable imaging conditions. Furthermore, our AI denoising feature will allow noise-free, quantitative analysis, enabling researchers to extract precise data from their imaging experiments. Now, based on this, I'd like to briefly introduce some of the immune-related research we have conducted using our intravital microscopy system. First, we observed high endothelial venules, as known as HEVs, the vascular structures that allow immune cells to enter lymph nodes. Using our system, we tracked how T cells and B cells migrated into the lymph node through the HEEVs. We also visualized fibroblastic reticular cells, as known as FRCs, to understand the internal lymph node architecture and how these cells guide immune cell movement. This was captured over a continuous 120 minutes observation period. Second, we studied neutrophil behavior in the lungs of sepsis model. By imaging the lungs of septic mice in real time, we saw red-labeled neutrophils accumulating and blocking green-labeled blood vessels over time. This obstruction created black dead space, which is a critical step in the development of sepsis. Third, we tracked bone marrow cells after transplantation. Over time, we could see the nuclei of bone marrow cells dividing, highlighted with yellow arrow here, as well as their movement toward the bone marrow sinusoids. Finally, we visualized an immune response in the skin. In the ear skin, we induced inflammation and tracked granulocytes moving toward regions marked by CARS protein. As time progressed, more and more granulocytes gathered around these protein-marked sites, illustrating the progression of the inflammatory responses. Next, I'd like to show you how our system can be used to study immune responses in cancer. First, we observed T-cell dynamics in real time within tumor tissue. We could see T cells traveling along the blood vessels, then extravasating, moving out of the vessels toward the tumor cells where they engaged in immune activity. Second, we tracked drug movement and biodistribution in tumors over a 24-hour period. Over time, the drug carriers traveled through the vasculature toward the tumor site. By the 24-hour mark, they had extravasated and accumulated around the tumor cells. While this particular study lasted only 1 day, our accessories allow long-term imaging, ranging from 2 weeks to an entire month. Finally, we visualized drug delivery in the liver in real time. As you can see here, nanoparticles move through the liver's vasculature, allowing us to monitor their path and distribution. This approach enables researchers working on drug development or immunotherapy to directly observe drug biodistribution and efficacy and immune cell dynamics in living tissue, which provides deeper insights and accelerates research progress. Beyond the applications I've just introduced, our intravital microscopy system can visualize a wide range of organs in living mice. For instance, we can monitor the brain, such as in brain tumor studies, and the small intestine and the kidney in real time. Additionally, imaging is also possible in skeletal muscles. In fact, when studying structures such as collagen or muscle fibers, our two-photon system can leverage second harmonic generation, as known as SHG, to visualize them without the need for any staining. This flexibility allows researchers to design experiments tailored to a variety of scientific goals and study requirements. Okay, then as you can see on this slide, research using I-beam technologies intravital microscopy has been continuously published in international journals, and this list is still growing. Compared to traditional tissue analysis methods, intravital microscopy offers a major advantage. It enables real-time observation at the single-cell level. This makes it possible to analyze fine biological mechanisms with greater clarity and quantitative precision. As a result, the technique earns strong trust from reviewers in both journals and conferences, thanks to the solid scientific evidence it provides. Now, I'll show you data where we used the intravital microscope without an imaging window to observe the real-time movement of immune cells over a short period. In this study, we used lyse-M GFP transgenic mice, which express GFP endogenously in neutrophils and macrophages. To clearly visualize the vascular structures, blood vessels were stained with CD31 antibodies labeled using our in vivo labeling kit. And we performed imaging on the mouse's ear skin, with maintaining the mouse's optimal physiological condition. As you can see in the video on the right side, the blood vessels appear clearly in red. The time lapse footage, captured at 30-second intervals, also shows the green labeled immune cells dynamically moving in and around the vessels. After confirming the dynamic movement of immune cells through ear skin imaging, we injected a fluorescently labeled specific protein drug of interest, which is the CARS protein, into the ear skin of the same lyse-M GFP mouse. CARS is a lysyl tRNA synthase, originally known as the housekeeping enzyme for protein synthesis, but more recently reported to act as an extracellular signaling molecule that modulates immune and inflammatory responses. We then performed imaging and analysis over time to observe inflammation induced by the drug injection, focusing on neutrophil dynamics. Imaging was conducted immediately after injection, as well as 3 hours and 6 hours post-injection. The results showed that over time, immune cells were actively recruited at increasing speeds to the site of protein drug injection, and they form clusters. This process was tracked in vivo in real time. Compared to the control model injected with BSA, we observed that immediately after drug injection, the CARS protein injected group showed a significantly higher number of immune cells being actively recruited and gathering at the injection site than the control group. As time passed, by 6 hours post-injection, some immune cells also gathered in the BSA-injected control group. However, in the CARS protein drug-injected group, a much larger numbers of immune cells had already accumulated at the drug site, forming large clusters. Additionally, we observed a significant increase in both cell mobility and velocity in this group. Next, I will show you an example of intravital imaging in the popliteal lymph node. Here, you see a mosaic image of entire popliteal lymph node, where red spot indicates CMTMR labeled T lymphocytes, and green represents CD31 labeled blood vessels. The plum-shaped endothelial cells forming the green color are called high endothelial venules, or HEVs. Specialized vessels that mediate lymphocytes' entry into the lymph node. After intravenously transferring T cells, we observed them entering the HEVs through multiple entrances connected to capillaries. You can see the T cell adhering to the endothelial cells and exhibiting prolonged rolling behavior, which is critical for their migration into the lymph node. So to be clear, I'll show real-time intravital imaging of the post-capillary HEVs in the palm-middle lymph node, focusing on the flow of adaptively transferred cells inside the blood vessels. As you can see in the video, red blood cells appear in blue and T lymphocytes appear in red. This imaging allowed us to observe the changing velocity of individual cells flowing through the vessels, as well as their spatial distribution. In the image on the right, the high endothelial venous, HEV, is outlined with a white dot line, as you can see here. Using a velocity color map, we visualize the flow speeds of both RBCs and T cells. We observed that both RBCs and T cells significantly slow down as they pass through the HEV compared to the other blood vessels. Notably, T cells adhered to the lumen of the HEV, rolling along and interacting closely with the high endothelial cells. This detailed tracking highlights the unique hemodynamic environment within HEVs that facilitates immune cell interactions. To better capture the full migration process, we used GFP expressing T cells, which is in green color, and labeled vascular endothelial cells with CD31, which is in red, and fibroblastic reticular cells, or FRCs, with ERTR7 antibodies, which is you can see in blue color. With this setup, we could clearly follow each stage of immune cell migration, from adhesion inside the blood vessel to crawling along the lumen, crossing the endothelium, moving through the perivascular channel, which is PVC, passing the FRC network, and finally entering the lymph node parenchyma. Through long-term 3D imaging of the same sites, we further discovered that lymphocyte extravasation does not occur randomly across the entire HEV. Instead, it happens repeatedly at particular regions, or so-called hotspots, which are specialized axis sites where immune cells preferentially leave the bloodstream. Identifying these hotspots provides important spatio-temporal insight into the migration dynamics of individual immune cells and shows how HEVs actively regulate where and how lymphocytes enter the lymph node. Next, we wanted to explore the relationship between dendritic cell, as known as DC, coverage of HEVs and lymphocyte extravasation in the lymph node. To do this, we performed real-time multicellular intravital imaging in living mice, where we could simultaneously visualize HEVs, fibroblastic reticular cells, T lymphocytes, and dendritic cells. A key question was whether T lymphocytes extravasate at sites where HEVs are covered by disease simply by chance, or whether there is a true preference for these disease-associated reasons. To address this, we compared the proportion of hotspots that were covered by disease, also referred to as trans-FRC migration sites, with the overall fraction of HEVs that were associated with DC coverage. Our analysis revealed that the proportion of hotspots overlapped with DC coverage was significantly higher than would be expected based on general coverage of HEVs by DCs. This finding clearly demonstrates perivascular disease around the LNHEVs are not just passively present, but are actively linked to regulating lymphocyte extravasation. Okay, so now I would like to introduce an example where we used our intravital microscopy system together with the dorsal skin fold chamber, to observe tumor infiltrating lymphocytes or TILs in real time within the tumor microenvironment. As you can see on this slide, we have developed a variety of accessories tailored to the characteristics of different organs. These accessories allows us to perform long-term imaging in living mice under stable conditions. For today's webinar, we used this dorsal skin fold chamber or DSC as highlighted here to carry out extended intravital imaging. Okay, so the first example I'd like to show is the immune response observed within cancer tissue. Using lysine GFP mice implanted with the DSC, we injected B16F10 cancer cells and tracked the immune reaction over time. On the day 1 after injection, we could already observe macrophages infiltrating toward the tumor site, and their coverage expanded progressively over time. In addition, macrophages accumulated around the cancer cells themselves, indicating an active immune response, while we also noticed morphological changes occurring in the surrounding blood vessels. We performed a similar experiment with MC38 cancer cells. Here as well, macrophage infiltration was evident from day 1. However, compared to the B16F10 model, the infiltration occurred across a broader area. As the response developed, macrophages increasingly surrounded the tumor cells, accompanied once again by noticeable vascular remodeling. The next experiment I'd like to introduce focuses on evaluating the degree of TIL, or tumor infiltrating lymphocytes, infiltration across 3 different breast cancer models. The first case was based on an allograft model where 41 cancer cells were implanted. To mimic adaptive T-cell therapy, we injected fluorescently labeled T-cells into the system. Using in vivo microscopy, we then tracked and visualized the activation state of TILs over time, specifically at day 1, 3, 8, and 11 after injection. On the left panel, you can see how TIL activation changes progressively over time. On the right, the time-lapse imaging demonstrates the dynamic process of T cells infiltrating into the tumor tissue. In addition, quantitative analysis of the imaging data shown in the lower graphs revealed a clear trend. As time went on, T cells became increasingly motile and more actively engaged with the tumor microenvironment. The next study was conducted using the same 41 model, but this time we aimed to evaluate whether there are differences in TIL activity between the tumor core and tumor periphery. With the help of in vivo microscopy, we were able to directly track and compare T cell dynamics inside versus outside of the tumor. As you can see in the left panel, T cells located inside the tumor displayed highly active behavior. In contrast, T cells at the tumor periphery showed more restricted motility. On the right side, the graphs present quantitative analysis, including track displacement length, mean velocity, with significant differences. Taken together, these results clearly demonstrate that T cells in the tumor core are more active and exhibit more dynamic responses compared to those in the periphery. In the previous slide, we compared T cell activity inside and outside the tumor and observed that by day 11, T cells activity within the tumor core had markedly increased. Building on those findings, I would now like to introduce results obtained from a xenograft model. In this experiment, we implanted the human breast cancer cell line MDA-MB231 into immunodeficient mice to establish tumors. This approach revealed a different pattern of T cell responses compared to the allograft model. In the xenograft setting, T cell infiltration and activation peaked around day 3 after adaptive transfer, but then gradually declined over time. This trend can be quantitatively confirmed in the graphs shown at the bottom side and is also visually apparent in the video on the right. So far, we have examined the infiltration patterns and functional dynamics of tumor-infiltrating lymphocytes, or TILs, through 2 complementary experiments. In the first experiment, using a syngenic 4T1 model, we were able to track TIL infiltration into tumors and monitor their activation over time. In the second experiment, we used xenograft model, MDA-MB231 and MCF7 to investigate how T cell responses differ depending on the tumor cell type. The slide you see here shows a direct comparison of TIL responses between the MDA-MB231 and MCF7 model. As previously reported, these 2 cell lines differ in estrogen receptor expression. In our study, we demonstrate for the first time that this difference can influence TIL motility and activation, as revealed by real-time imaging and quantitative analysis. In fact, T cells in the MCF7 model exhibited higher motility and activation levels, which is also statistically significant. Taken together, these results highlight that in vivo microscopy is a powerful approach to dynamically track TILs and tumor cells, providing real-time insights into the interactions between the immune system and tumors. That concludes my presentation for today. Thank you all so much for your time and attention. I hope the data I shared demonstrating how intravital microscopy can be used to observe immune cell dynamics in various models has been helpful. Thank you again for joining us, and I'll now hand it back to Morgan.
Thank you so much for the great presentation. Before we conclude, I would like to mention that we are currently accepting applications for on-site demonstration at our demo centers in Europe. I will take a moment to share this information, and in the meantime, please feel free to type any question you may have into the Q&A box below. In Europe, our demo location includes VIB, Leuven Cancer Biology Center in Belgium, and CRBS in France, and University of Tübingen in Germany. These sessions offer a valuable opportunity to gain hands-on experience and training in intravital in vivo imaging in live animals. Now, let's begin the Q&A session. So, our first question is, how long does the fluorescence signal last when you label the blood vessels?
Okay, so as you know, in vivo experiments inherently involve many variables, so signal duration can vary. Typically, the fluorescence signal can be maintained for about 1 to 2 days.
Okay. The second question is, can this imaging approach be applied only to small animals such as mice?
Okay, for that question, not at all. We also have models for larger animals such as rats and parrots and etc. So using our free space system designed for medium-sized animals. So therefore, you can choose the appropriate equipment based on the animal size you plan to image.
And the third question is, once you implant the chamber, how long can it be kept in place?
Okay, so we recommend performing the surgery in as clean and sterile environment as possible, ideally within a clean bench. So with proper post-operative care, so including inflammation management and monitoring the animal's condition. So the chamber can be maintained for long-term imaging for up to around 2 months.
Oh, okay. So, thank you everyone for your questions. The webinar recording will soon be available on our YouTube channel. So if you have any more questions or if we couldn't address your inquiry during the Q&A session, please feel free to reach out to us at information@ibeamtech.com. And as I mentioned earlier, we have recently launched the iBeam shop, where you can find imaging accessory, fluorescent dyes, and training programs. By registering an account, you can access certain benefits applicable to your purchases. Thanks again for joining us, and we'll look forward to seeing you at our next webinar. Thank you.